International Bat Week. Got it. Happy International Bat Week. It is one of my favorite times of the year. I love Halloween season. I love International Bat Week. So to go with Laurel's question about questions, totally fine. If people want to ask questions, chat. I will be monitoring the chat. I love seeing chats when I'm giving presentations. I love seeing reactions. So feel free to put, if you have any questions while I'm talking, you can put them in the chat and I'll try to address them on the spot if I can. Any longer questions, we should have plenty of time at the end for that. So let me get my screen situated here so I can see the chat today. And I think we have some people whose microphones might still be unmuted. So let's try to make sure we're muted there. But let's just kind of kick this off with a little bit of kind of what I plan on talking with you guys about today. So fundamentally, as bioacousticians, our heart is in this for conservation. So conservation is at the heart of what we do. I'm going to imagine that most of you are, like me, are using acoustics as a tool to understand animals so that ultimately we can help protect them. So we're understanding populations and distribution and behavior. So today I'm going to be sharing with you some of these acoustic tools, acoustic methods that I use to help understand the population size of really dense aggregations of single species of animals. So I'm going to be telling kind of my story today in 3 acts. We're going to break this into the past, the present, and the future. So I'm going to be talking about my history into the development of the methods I use. I'm going to be talking about some kind of in the moment now research that's happening, some of the really exciting pilot data that we're getting. And we're also going to, think a little bit to the future and talk about what can be and how we can use some of this acoustic methodology combined with emerging technology for really the next frontier of animal monitoring and species conservation. And I'm really looking forward as well to questions from you, as well as hopefully a nice discussion that we can have at the end of this. So before we begin, let's kind of put aside our scientific brains for right now, and let's go on a little bit of an imaginative journey. So I want you all to imagine with me. I want you to imagine a future in which, much like this map of migration information of birds that we get from visual observations, I want you to imagine a future in which we can get this type of information, but not from visual observations, but from acoustic data, and that we can get this type of information in real time. So imagine a future in which we can get real-time information on the migration of an animal. And for the beginning part of my talk, I'm going to use a bat as the model organism for this. So I want you also to imagine a future in which, by monitoring a single bat roost or a bat cave, we can not only get information on the population at that bat cave, but we can also get information on the birth rates at that bat cave. And furthermore, information on the dispersal of where those bats are going to other bat caves. And then let's take that one step further. And by imagining a system in which we monitor networks of caves that are close to each other or more than one cave, we can get species-wide information on information such as the timing of migration for migratory bats or the timing of hibernation, how bats are responding to climate change, as well as identifying at-risk locations for conservation. So the goal with what I want to talk about today is hopefully to convince you that we don't really need to imagine this future, because we actually have everything in our toolbox right now, and the technology exists to do this. But the technology exists to use passive acoustics and autonomous monitoring in order to get real-time population information on a single species. And in this case, we're going to begin with bats because bats are what I've historically studied, and the bats are kind of the framework for this. And so all of this is focusing on a method that I'm calling censusing via acoustic energy, or CVAE, meaning counting animals, in this case bats, with sound. But that doesn't sound that exciting because most of us are doing this anyways. So it's really important that I take a moment to explain in order to help you understand what this method is, it's important to help you understand what this method is not. So first of all, this method is not detecting individual calls or individual bat passes. The historic way that we approach monitoring bats with acoustics is by having a single acoustic recorder, and then the bats are flying overhead, and we get these bat passes, which are multiple echolocation signals of a bat passing overhead. Here's a spectrogram of what this looks like. You can see the individual FM pulses of a single bat passing over. And then, like many animals, bats have species-specific vocalizations. So from these recordings, you can get species information. You can then, depending on comparing habitat types, you can get presence-absence information, occupancy modeling, and even you can estimate relative activity levels. But again, my method doesn't do this. This is not what my method does. Okay. Another important distinction of what my method is not, what this method is not, is this method is not using any sort of machine learning. Okay. I love machine learning. It has fantastic applications. I use it all the time for the other arm of my research, which is trying to understand the echolocation behavior of bats in medium density groups, or trying to identify species from mixed aggregations. Machine learning, we all know it. We all hopefully love it. Many of us probably use it. It has wonderful applications in the field of bioacoustics, but it also can be computationally intensive. This method, the censusing via acoustic energy, does not do this. All right, so let's go into a little bit of specifics as to some of the characteristics of what exactly this method is. Well, is not and also is. So again, the CVAE, censusing via acoustic energy, it does not use machine learning, does not require identifying or extracting individual vocalizations, and it's not using any sort of acoustic indices as a measure of biodiversity, because this method is designed for dense aggregations of single species of animals. So we know there's a single species that are there. We want to try to estimate the number of individuals of that single species. We do not need, or we cannot physically separate individual vocalizations. So one of the benefits is that this approach uses very little computational power. It's just a couple lines of code, which lends itself very nicely for true autonomous sensing that I'm going to touch on in just a little bit. So I mentioned that I primarily study bats until I kind of took a side journey in my research into this direction of using acoustics for population censusing. The main thrust of my research was understanding sensory motor behavior. Did my PhD work with dolphins with a sample size of 1. Now I worked then in my postdoc, and then the beginning of my career switched over to bats with sample sizes of 1 million plus. So with these big aggregations of bats, extracting individual vocalizations is a challenge. So I'm going to play a video first, and I'm going to kind of narrate over the video because I could never get tired of watching bats fly across the screen. So you're looking at an emerging colony of the Mexican free-tail bat, also called the Brazilian free-tail bat, Tiderata brasiliensis. This is thought to be the largest mammalian migration on the planet. We don't exactly know how many bats are migrating, but we do know that the bats are migrating up from Mexico and Central America to discrete cave roosts in the United States. Females form these really large, dramatic maternal colonies. If any of you have been to some of these bat caves or seen a big bat cave emergence, I'd love to hear it. Love to let me know in the chat. It's always nice for me to get a pulse on how many people have seen a big bat emergence. But the females form these really large cave colonies where they come pregnant, they give birth inside the cave, they raise their pups, and then everybody migrates back away. So these are habitats with a lot of bats that leave all at once. Okay, so we've set the groundwork for this. I also mentioned that it's thought to be the largest mammalian migration on the planet, but we actually know, it blows my mind how little we actually know about the populations of bats. And there's some reasons for that I'm going to come in just a minute. So someone asked in the chat, where are the males? The males form smaller bachelor colonies. They do migrate up, smaller bachelor colonies, but these big caves are predominantly females. Yes, and the females are pregnant when migrating. So this is an acoustic methods seminar. Let's talk about the acoustics of this. So you can see a spectrogram of a 250 millisecond portion of this emergence below, which I like to call acoustic spaghetti because it's very complex to try to tease out the individual vocalizations. Let's listen to what this would sound like if we slowed this down to 10% its original speed, because bats are ultrasonic. So, stop that there. A very complex and chaotic environment. How on earth can we use acoustics if, oops, I lost my slides here. How on earth can we use acoustics, if at all, to count these bats? Well, first of all, let's talk about some of the motivation for counting these bats, period. So we mentioned that we know very little about bat populations worldwide, even in the United States with all the research effort. But it's really important that we have accurate populations for a couple reasons. So first, in the United States, there's a pathogenic fungus called white nose syndrome. Well, the pathogenic fungus results in white nose syndrome. For those of you who are unfamiliar with this, this is a pathogenic fungus that is native in the caves in Europe. Bats have immunity to this or resistance to it. It was introduced, it's thought that it was accidentally introduced to upstate New York at the winter of 2006, 2007, and it has been decimating bat populations across the US of the hibernating bats. The hibernating bats are vulnerable to this. We know that in some caves where they have been monitored, some populations have up to a 99% decline. But because of the challenges with population monitoring, we actually don't fully understand how these populations are being affected because we have a lack of data on population in many of the bat caves. It's also really important to get accurate population estimates to inform the listing or the delisting of species. So here in the United States, we're talking either threatened or endangered species that get different levels of protection from either the state or the federal government, depending on their population size and how those populations are changing. And then further, it's really important to get population estimates to identify priority conservation areas and also to understand the impacts of conservation efforts. So for instance, sometimes we put gates on cave openings as a way to prevent humans from entering and disturbing the bat colonies. We don't fully understand, we have a little bit of information as to how that affects the bat populations, either positively or negatively, but we lack a lot of information as to exactly that effect. So historically, the way that bats have been counted can kind of fall into three buckets: visual estimates, mark recapture sampling, and thermal imagery. But each of these historical estimates has a disadvantage. Visual estimates can be really prone to bias. Mark recapture sampling can be invasive, especially when we're talking about the pathogenic fungus, and time consuming. And thermal imagery is really emerging kind of as the go-to for monitoring bats. But even with technology becoming more affordable, it still requires fairly expensive equipment. And for many of the stakeholders I work with, which are some of the state conservation agencies, they just don't have it in their budget to afford all the equipment necessary to do these surveys. And until fairly recently, counting of these videos was done manually, which when you're talking, thousands and thousands of bats, that is incredibly time consuming. So the motivation for this was to see if we could use acoustics as a way to estimate the population of bats. So let me walk you through the framework for this. And from a methods perspective, I think this is actually really simple. I mean, I could have just come in here for 5 minutes and talked about the actual math and the method behind this acoustics, but my talk today is more about how we can put this methodology into practice. So this has been a really fun side project that began as kind of just like this fun observation I saw in the field, and then we published on it several years ago. But as I mentioned at the beginning of my talk, this has really been a new side direction in my research, which is great and it's exciting. And so depending on where you are in your career, you never know what side project might guide your career path. So keep an open mind with that. But the framework for this is pretty straightforward. The framework for this requires having video recordings and acoustic recordings that are synchronized together. So the general concept is that when bats are leaving from a cave, you have a video camera or a thermal camera And then you have some sort of full spectrum audio recorder. And you can place this either just outside the cave. We also place this just inside the cave. But the idea is that you're capturing this volume of bats that are leaving from a known location. And so if you look at the number of bats that leave over time, so most of these bat emergences, depending on the colony size, can last anywhere from 15 to 90 minutes a night. And sometimes you have multiple emergences a night, depending on where you are in the season. But there is a very specific beginning period and a very specific ending period. So at the top, what I have is just an example of what an emergence profile would look like if you were recording with the camera. So as the emergence begins, the number of bats leaving at a given time window increases, and you can see the corresponding change over time. And then you can also relate this to the acoustic amplitude. And so we were finding, the concept is that more bats, more acoustic energy, right? So if you take this long emergence and then you break it up into discrete time windows. In our case, for the first stab at this in the publication, we did 1 second time windows. The theory is that there should be a relationship between the number of bats leaving in a given time window and then the sound level in that same time window, okay? So we explored this with 2 different caves that were located very close to one another. These are still the free-tailed bats that I was talking about. And at the first stab at approaching this, we evaluated 3 different metrics for calculating the acoustic energy. And I mentioned this is really, really straightforward because all we're doing is a high-pass filter. Other than that, we're not doing any other signal processing with the data. And we're doing everything basically on the level of the waveform. So the 3 metrics we evaluated was RMS pressure, peak to peak pressure, and then the total energy. And we found that there was a significant relationship between the number of bats leaving in a time window and all of these 3 parameters for calculating acoustic energy, but we had the best fit with the RMS pressure. So you can see the data for the two different caves. Each color represents a different cave location on the graph here. If you pay attention, the y-axis is on a log axis, or well, not on a log axis, but the data have been transformed to the log count. So if you kind of do the math, we're talking a lot of bats. So this is a very, very high density situation that we were evaluating this at the beginning. Really strong relationship for high density bat emergences. We also found that the best performance of the CVAE occurred when you average it across multiple nights. Okay, so let's talk about the equipment. That's part of methodology, right? What's some of the equipment that we use? I lost my mouse, here we go. So in my lab, I use a lot of different equipment. I mentioned one whole other arm of my research is sensory motor behavior of bats. So we use, because bats leave sometimes, depending on the location, when there's sufficient ambient light, we use standard video cameras. We also use thermal video cameras. We have played around with a lot of different camera models. There's a sampling of some of the ones we've used here. The good news is it really doesn't matter as long as you have a camera that is sufficient to get some sort of ground truthing on the number of bats. That's all you need. In terms of the acoustic equipment, the only thing you need is to make sure you have a full spectrum continuous acoustic recorder. So it has to be able to record continuously. We use audio moths a lot. Before audio moths, we used SM3s. Some of the newer wildlife acoustics, like the mini and the micro, those can't do continuous recordings. You're limited by a window, so those aren't very useful. But we're kind of moving in the direction of using audio moths. We've been using them since they first came out, and they work very well for this application. And they're also incredibly low cost. And because they're open source, they're easily modifiable for adding on other components, which I'll talk about in just a second. So I mentioned that the framework that I just introduced requires having this known count of the number of bats. And when we first began, we were doing this manually, and it took forever. I had so many students helping me count bats, and it was like, there has to be a better way. There has to be a better way. And we found out that there used to be some software programs that existed through Fish and Wildlife, but they'd kind of been phased out because they were with antiquated systems and you couldn't use new technology. So we were motivated by that, and we made our own counting software. which has been a really fun outcome of this project. So here's just an example of what this software looks like. This is a thermal recording taken from inside of a cave. You can see the bats moving around. This is actually during emergence. So you have some bats that are, bats sometimes will come back in and do a couple loops before they go out. But this software basically allows the user to select specific regions of the video, and then you can set different threshold values, different pixel sizes, and it just detects the objects and it counts them as they move across whatever box the region defines. So here you can see the user wanted to, count bats in 2 regions, so the blue region and the purple region, and it counts bats as it crosses through those regions. And I mentioned that sometimes we have bats that go back and forth. So in this situation, it's counting bats as they're going each direction. This has been a really fun process to work on this software. I don't get the credit for doing this at all. That's not in my wheelhouse. I have a brilliant, he's actually a physicist who's been writing the software for us, but it's been really fun to develop because we did this in incredible close collaboration with the people who are actually going to be using it, the Fish and Wildlife Service officers. So the different people working in the state offices, they were the ones that actually informed us on the features in the software and helped with all the beta testing on this. So it's been a really fun project. And the benefit is now I get to count bats a whole lot faster. So that bottleneck I had of trying to count bats, that's out the window. So I have this data set that has been piling up since 2016. I have so many recordings at bat caves, and now we're just running our videos through our software to get the counts. And we're working now on doing the relationship between the acoustic energy to figure out how well this is performing across location. So let me just give you an example of some additional validation data we have so far for CVAE. The good news is that we tested this at caves that have that are not as dense as what I introduced in a few slides ago from the initial paper that described this process. We've been testing this at lower density caves. We've been testing this at different locations, different cave types. We've also tested this with a different species of bat, a gray bat, which is a hibernating bat that doesn't form as dense of caves necessarily as the Brazilian free-tailed bat. And what's great is that we're still finding a really nice relationship between the sound level, and we're still finding that RMS amplitude is the best measure. So we're finding consistently RMS is performing better than peak to peak or the total or the sum of the acoustic energy. And we still have a little bit of noise or uncertainty in that population estimation for the really low dense bat emergences. As you can see here for the gray bats, right? We just have a lot of spread around our data points. But again, if we sum that up across an entire emergence, if we take those individual data points, we're finding that the acoustic estimate, the acoustic population estimate, can be really accurate at estimating the total population compared to video. So one of our test cases, we found 96% accuracy between the acoustic estimate and the video counts. Okay, so now that was the past. Let's move into kind of thinking about some of the future with this. Where can we go from here? Why is this important that we can do this? Well, I mentioned that computationally, this is very simple to do. So imagine a situation where if we know, I'm going to go back for a slide. Imagine, let's say this Missouri cave here, this gray bat cave in Missouri. We know what this equation is that relates the sound level in a time window to the number of bats per second. So imagine if we know what that equation is for a cave, in the future, all we have to do to get a population estimate is have an acoustic recorder that's doing that calculation, and then it can spit out the overall population for that cave each night. Now, some of you might be thinking, okay, Laura, that sounds perfect and idyllic, but is that really gonna, how stable is that really gonna be? Well, the answer is we're currently figuring that out. So we're in a deep dive, again, with our Fish and Wildlife partners. We're currently trying to figure out for a given cave site, how stable, if we figure out this regression equation for a given night, how stable is that over repeated nights? Theoretically, if you have the same recorder and it's at the same location, the only thing that should be affecting that is temperature and humidity. But we all do field work, right? We know that theoretically that's not going to be possible. So we're trying to figure out how much uncertainty is there. We're also trying to figure out if we move between sites or use different acoustic recorders, what is the influence of all of those parameters on that regression equation? Because if we can figure that out, we can then move towards kind of this all-encompassing acoustic model where down the line, people could just skip this video ground truthing entirely, make the acoustic recording, input the relevant metadata. So what type of acoustic recorder you're using, what's your gain setting, what's the distance from the recorder to the bats, what's the species of bats, the temperature, the humidity, any other physical parameters that might describe your environment. And then you could get a population estimate plus this uncertainty. And I'm putting my hands up like this because this is the stage where we're at. What exactly is this uncertainty? And I think it's important to talk about this uncertainty for a little bit because this is a trade-off that we have with acoustic monitoring. So we have this trade-off in that acoustic monitoring can save us an incredible amount of time and an incredible amount of cost, but we're always going to have a little bit of uncertainty in our estimate. And so what level of uncertainty are we willing to take? And when I say we, that really depends on who you're talking to. So if you're talking to a researcher, I would say I don't want a large number of uncertainty because I'm trying to get, I need a very accurate estimate because I'm trying to do something statistically that needs this p-value and, we need as accurate as possible. But if you're talking to a stakeholder like Fish and Wildlife that are often monitoring caves that they have to hike 5 miles in to access a cave, they have one thermal camera that they're rotating throughout their whole state, and they have a one-time shot to get that population estimate, they might be willing to have a lot more, to tolerate a lot more uncertainty in that population estimate if it means it can be automated and they can get this information every night. So we're having this conversation with the stakeholders, trying to figure out what level of uncertainty is good enough. And we're also trying to figure out if we can narrow the uncertainty in our population estimate with additional signal processing approaches. So we're, like I mentioned before, we're really kind of in the thick of it right now, just swamped with data, trying to run all of these values through and trying to figure out the model and the uncertainty in that. Because if we can figure this out and we can get this to what is considered an acceptable uncertainty in the population estimate, this is where we can truly have autonomous monitoring. So we could have a system where we use an audio moth that I mentioned is open source, so you can modify it, you can power it with solar power, you can have it sending 1-second snippets of acoustic data to a data logger or a microcomputer that's doing that population count within that window, pinging out every 1 second, oh, here's the estimate of the bats in this time window, sending that to an autonomous sensor that we can then get real-time population data. And the exciting part is that all of the hardware we need for this exists. We've spec'd this all out. We have a couple irons in the fire in terms of funding. We're hoping will pan out so that we can set up this first pilot sensor to basically try this at one of the caves where we found really strong agreement, really strong accuracy with the CVAE. We're hoping to get this set up in the next year or so to get this pilot sensor, because as I mentioned before, we can then move to looking at range-wide population estimates. So there's a reason why I'm focusing on the Brazilian free-tailed bat, the gray bat. So it's estimated that about 90% of the migratory population of the Brazilian free-tailed bat goes to 21 discrete cave roosts across the Southwest. So theoretically, we could put 21 sensors and we could monitor up to 90% of an entire species. So imagine how powerful that could be if we could get real-time information on when they're migrating to the different caves, as well as range-wide population information for almost this entire species. And by doing that, by getting this information from this network of caves, we can do more powerful things when it comes to ecology and population biology. So for example, I collaborate with some really brilliant mathematical modelers, and they're starting to kind of set the framework for how we could make predictions about dispersal. If, say, one of these key bat caves, if something happens, like an earthquake or a fire or whatever, habitat destruction that caused that to be unsuitable habitat for those bats, how would that affect the other bat caves? And would that actually have an effect on the species? Would the other bat caves be able to absorb that population, or would it cause a mortality in the species in general? And now here's where it gets really exciting. I haven't yet talked about phenology, but we're going to talk about phenology. So from the emerging population, we can even have the potential to pull out the timing of key life events for these bats. So I mentioned that these are a migratory species. The females migrate up from Mexico and Central America. They have their baby bats. The baby bats are born, and then they all migrate back. So let me walk you through this illustration here that highlights some of the key phases in the life cycle of the free-tailed bat. So in time period 1, this is when the population would be migrating to a cave. So imagine that this is showing a graph of just if we had that acoustic recorder recording outside the bat cave each night and what that population would be. So the nightly population of bats leaving the cave. So as the bats start to migrate to the cave, you'll see those numbers go up and up and up. Now, time period 2 is very critical because I mentioned these bats come pregnant and then they give birth inside the cave. So I think almost all insectivorous bats, if someone knows differently, you can put it in the chat. They don't carry their baby bats with them. They leave them behind. So the free-tailed bats and the gray bats, they absolutely do. They leave their pups, what we call baby bats, on the cave ceiling. And so in time period 2, those females, we would not predict them to disperse anywhere else because they have to come back each night to nurse their offspring. So they're not going to disperse. We're assuming constant, of course, there would be some mortality with, just general mortality from predators when they're flying. But time period 2 would represent the stable adult population of females for that year at that cave. Now, for the first 6 weeks or so of their life, the juveniles can't fly, but once they begin to fly on their own, we call that becoming volant or achieving volancy, which is a fun vocab term. We would start to see that emerging population increase. Time period 4 would be when those baby bats are all flying with the mothers. So by comparing time period 4 to time period 2, we can theoretically get information on birth rates of the cave. And then time period 5 is when they migrate back. So just by looking at the emerging population, you can extract all of this information on the life history of these animals. So from there, we can also, by combining across the networks, we can then look for the entire species for these life history characteristics. Again, we talked about phenology and then how the species as a whole is responding to climate change. Now, I mentioned that I also study gray bats. I use gray bats as a model for this system, for this method, because just like the free-tailed bats, for the gray bats, which are a hibernating species, an endangered hibernating species, it's been estimated that about 90% of their population hibernates in 15 discrete caves. So we have these 2 bats with a huge proportion of their population in really discrete locations. So these lend themselves very nicely for evaluating this method. And we can even kind of think more broad scale about expanding this up to moving beyond population counts for just general bat activity levels, especially around wind turbines. We know that wind turbines cause a lot of bat mortality, but imagine if you could network sensors across a wider area for smarter curtailment or real-time monitoring to have almost real-time information on when to shut down those wind turbines. So there's some additional directions that those can go, that it can go beyond just population counts. Okay, so those were bats. It is International Bat Week, but I did promise in my title that we're going to be talking about more than just bats. So now let's shift gears, and this is definitely more of like in the future because these are all projects that are in different stages of development, but very much beginning stages of development. We're going to be talking about expanding this method to other single species aggregations. Okay, so frogs, I'm going to be talking about frogs and penguins, specifically with frogs, the bullfrog, the American bullfrog. So the American bullfrog is native to Eastern North America, and it's pretty ubiquitous in aquatic environments. Unfortunately, globally, it's considered an invasive species. And when it's introduced into habitats, it can outcompete the native amphibians. So as a result, trying to understand where these frogs are and their populations is really important for native species conservation and for just general management of ecosystems. So The good news for us bioacousticians is that bullfrogs are highly vocal, incredibly vocal. So during the mating season, male bullfrogs call out to emit advertisement calls to females, and they also call out to defend their territories. So the result is that you get these frogs that are calling back and forth to one another, and we call this a chorus. So this results in if you have a lot of frogs in an area, a really complex soundscape of overlapping calls when there are multiple individuals in a pond. So this is a spectrogram below that shows, just from a simple case of just a few bullfrogs that are overlapping, what the sound file actually looks like if we were to graph it. But now let's take a listen to the beautiful chorus of the male bullfrog. So even just from a few individuals, you can hear how it could be problematic to extract those calls. So I was talking about this method that I was doing with bats with a collaborator of mine, and she said to me, "Oh, well, we should do this with frogs." And I said, "I'm a mammologist. I know nothing about frogs." And she has opened my eyes up to the wide world of amphibians. But here is what I have learned as to why these are great candidate species. So the behavior of bullfrogs lends itself very well to exploring this method, because they tend to hang out on the perimeter of ponds. So if you had a pond that is somewhat circular, right? There's a lot of assumptions we make in field biology. Theoretically, if you had I see a lot of great questions coming in. I will get to all of these at the end. I promise I will get to them all. If we have a microphone in the middle of a pond, theoretically, those bullfrogs should be roughly the same distance away, which is kind of important at this stage of where the CVA is that we have everything relatively the same distance. So she said, "Hey, maybe this could actually work for frogs." And I said, "Cool, I don't do frogs." And she's like, "Well, I have a data set of opportunity. Let's explore this." And so we did that this past year. We explored that. We kind of did a little pilot study where we looked to see if we could estimate the chorusing activity by using this acoustic energy. And the answer is meh. The answer is for 2 sites, for 2 nights, we found a significant relationship between, now, her study, I mentioned this was a historic data set she had, so historic recordings. She didn't actually count individuals, but we had the number of notes that we could manually count just by listening. You could actually have a trained observer listen and count the number of notes in very long time windows. I think this was like 5-minute windows we were using. But 2 of the nights, we found a significant relationship between this total acoustic energy and then the number of notes. 2 nights, we did not find a significant relationship. Those 2 nights also had a large amounts of anthropogenic noise. There were airplanes flying overhead. So the jury's kind of out as to if this will actually work for bullfrogs. But the exciting news is we're going to find out this summer, because we have some funding and we're going to do a deep dive around the UNH campus. There are 67 potential bullfrog habitat sites. I had I'm a very ambitious student, map this out for me. We identified 67 candidate sites. And so I have funding for undergraduates this summer to help join a project to survey all of these frog ponds. So you can count frogs by shining flashlights. We're going to be making the acoustic recordings and getting that actual count. So if you are an undergraduate, or if you know of undergraduates interested in paid research opportunities for this summer, contact me. You'll see my website at the end of my presentation today. Okay, and so finally, let's touch on penguins. This is going to be pretty brief because we haven't even started this yet. But let's talk about penguins. So the ADÉLIE penguin is located only on the Antarctic continent, and it is considered an indicator species because it is ice obligate. It needs ice for its life cycle. And it is highly vulnerable to climate change. These populations are being wiped out, but they are highly vocal. They are highly vocal at distinct phases of their life. So they form these dense colonies, the calls overlap. There's males that are calling out, there's pairs that are calling out. When the chicks are born, they start having their own vocalizations. You can see one of the complex spectrograms of a recording from inside a penguin colony below. Now let's listen to the beautiful soundscape of a penguin colony. Beautiful, isn't it? I love listening to those sounds. Okay, so when I was trying to think of how we could potentially scale up this idea to test out how this performs with other taxa, seabirds came to mind. And I met Megan Semino, who is from NOAA and UC Santa Cruz. And she says, this is fantastic. We go down to Antarctica every year. We count these penguins. We know exactly where these colonies are. They have very stable colonies. These colonies are stable from year to year to year. Once the chicks are born, they're not moving. They're not going anywhere. I have technicians that are counting these penguins all the time, taking detailed notes, mapping the colony sites. They know exactly when the chicks are being born and how many. I said, this is like a dream for me. Can I please send some acoustic recorders down? And she said, absolutely, sure. So we have all of our acoustic recorders that I think they should have just arrived in Antarctica, but we're going to be recording throughout the nesting season to try to understand how well, if we can use CVAE to estimate penguin populations. But more importantly, what we're really excited about is diving a little bit deeper. Oh, there's a pun in that because it has to do with their foraging behavior. Diving a little bit deeper into the characteristics of the recordings to see if we can use this concept of CVAE combined with some spectral analysis to understand foraging and phenology of the chicks or of the penguins themselves. So what I learned from Megan, again, I learned lots of wonderful things from my collaborators, is that penguins go on these incredible foraging trips. They can go on trips that can last for several days, and they can travel hundreds and hundreds of kilometers. And according to Megan, often a huge proportion of the colony will leave. So she said, oh, well, one of the really interesting side effects is maybe we could actually, if we could see a change, a theoretical change in the colony population from acoustics, we could use this as an estimate of how long they're foraging. And the foraging effort of penguins reflects the available food supply, which just plays into the whole concept of understanding the Antarctic ecosystem. So I mentioned that this is very much in the future, but the plan that we are going to begin in the next 2 weeks is pairing acoustic recordings with colony counts and ground-based observations so that we can determine the link of acoustic energy to the numbers of bats, but also maybe exploring some ideas of some acoustic indices. And I'm kind of saying maybe because I have a graduate position available in the fall to begin this. So I don't know which graduate student will be joining, but we have this is going to be a graduate student's project to investigate this. So I don't have the set plan exactly for how that will go. But we're also going to be kind of exploring some new avenues. We're going to be pairing with data loggers of microphones at different heights to see if we can investigate the effect of any thermal inversion, which can happen often in this environment, to see how that might affect CVAE. Because when we were talking about bat caves or frogs, it was a very simple, kind of a simple environment. But when you're talking about these colder climates, you can have really significant thermal inversions that theoretically could investigate the model. And what's really exciting is that just this month, someone else has already kind of laid the groundwork for this, in that they were looking at the vocal activity rate and found that by counting individual vocalizations, this can accurately estimate the penguin population, at least the chick population within a colony. So we're going to kind of take this one step further, looking at this concept of acoustic energy, not just for chicks, but also for the adults as well. So that wraps up our little journey today of the past, the present, and the future. So hopefully I have convinced you, or at least made you start to think about the possibility that we can do this for a wide variety of taxa. We have the technology to do this. We have the validation data, at least really strong for bats, meh for frogs, for penguins, I don't know, to conduct autonomous real-time population monitoring. But we can use this for species abundance, distribution, and phenology. So you can start to think about other dense aggregations of species that this might be valuable for. Other marine mammals like sea lions that like to haul out in large groups. You know, you can start to think, and I saw some comments coming through of people saying, could it work for this? Could it work for this? But this is kind of the general concept and kind of the new direction that we're going with. So I just also want to do another shout out that I am recruiting students. So I have an opening for at least one graduate student starting next fall, as well as undergrads this summer. So you can find information on my website on how, if you want to learn more about my lab. And with that, I think we will move on to the questions. Laurel, do you want me just to work through the chat? How do you prefer to do questions?
Yeah, that works for me.
Okay. All right, I see some, I'm not going to read out the comments. I appreciate the comments. I'm reading all of these out, but, can you tell different species apart? So Marcelo asked, can you tell different species apart? Yes. Marcelo, bat species have species specific calls. When you start talking about the myota species though, which is like, there's a lot that are very similar. So for the most part, many bat species can be identified by their calls. And so I'll show a slide. I didn't really go into details of it. I kind of like teased you with the slide. I did have a graduate student who was investigating if with very simple machine learning and clustering approaches, you can extract, when you have overlapping calls of mixed species cave emergences, pull that out. And so that is like, here's where I am with evaluating the model. I think the next step would be really trying to combine machine learning with CVAE to see if we can estimate the population of mixed species roots and parsing that out by species. Great question. Oh, okay. Do you think this acoustic method could work to estimate the population size of great amounts of phyllostomid bats? Phyllostomid bats have different echolocation parameters in contrast to aerial insectivore bats. Still, if many phyllostomid bats are emerging from a cave, maybe the amount of sound level per second generated from them could be measured, right? Absolutely. I have to say I'm not super familiar with phylostomids, but I think that this general concept, I mean, it's not just for FM bats. It's not, I mean, we're starting to find this concept can work for we mentioned frogs already. So absolutely. And I should also say, I should have made this very clear. If there are any of you that are already just like making acoustic recordings at bat caves and maybe are also videoing, if you want to collaborate on this, I'm always looking. I'm looking for people that want to help contribute to the data set. So please reach out to me as well. Let me put my slide back up here so everybody has my contact information. Because I so far have been very US centric with my approach, and I would really like to expand to see how different species of bats, especially at some of the really great bat caves, how well this might work at some of those locations. And Gisela said maybe in the future, I know Gisela. Yes, I hope I can come with you and we can record sea lions together. That would be lovely. Oh, Mitch asked, do you think this could work for insects? I don't know enough about insects, but if they didn't move, yes. Laurel, do you have something? Do you have anything, any insight to add with that?
Yeah, it depends a little bit on the community. We've been thinking about some of these questions, but if you have a community with very few species and they're frequency segregated, yes, a lot of times you get communities that are with a few species in the same frequency band and very different call rates between the species. And so I think it's doable, but it takes a little bit of parameterization.
Sure. Yeah. Okay. Awesome. See, that's exciting. Very cool. Okay. Can this method help to estimate the distance from the recorder to the source of the sound? So I think absolutely. It's not, I mean, we know from traditional acoustic analysis that you can, if you know the source level of a vocalizing animal, you could theoretically determine that. I think that, I mean, so one idea we have with the penguin is we're putting multiple sensors around the penguin colonies themselves, because if you think about it, with the frogs, all the frogs are at a known distance away from the microphone. The bats, all the bats are at the same distance away from the microphone. The penguin colony is going to be a lot different, because regardless of if we put our acoustic recorder in the middle or the edge of the colony, which we're doing, you know, one individual that's vocalizing right next to the recorder is going to be a lot louder than one that's at a different distance away. So we're thinking of some potential, we're not really sure. This is the straight answer. I like to be really transparent, and my work. A lot of it is like, I want to try to do this and let's figure this out. But we're exploring with some ideas of potentially normalizing amplitudes of vocalizations. We're also planning at some penguin colony locations of having multiple sensors, so potentially interpolating the data across the field, which is what people do when they're just trying to estimate, trying to get information on kind of single individuals that are vocalizing. But I think the answer is yes. We just haven't done it yet. That's a great question. Dimitri asks, I wonder if you could train a machine learning model to do a similar thing, and then you could add extra metadata as well. That's a really good idea. Sure. Yeah. If you want to send me an email, let's talk about that. Yeah, that's a great idea. See, I love all these comments I get. If this method works for frogs, and it will work for marine mammals as well, isn't it? Yes, it should. So we've thought about haul out sites for sea lions, for some of the really large aggregations of marine mammals. I mean, people are already doing density estimation with acoustics, like some of the traditional density estimation approaches, people are already doing it. But using this acoustic energy is kind of a simpler way, a way to step back from more complicated localization or more of the complicated machine learning approaches. Theoretically, it should, but again, I think the limitation, the reason why we haven't been able to put this into practice is that we need a recording, but we also need to know exactly how many animals are there for that ground truthing. And it's that ground truthing that really is kind of the crux of how to actually estimate if this is valuable. And I want to make very clear that there's absolutely going to be situations in which this doesn't work. There's going to be caves where this does not work. There's going to be taxa where this does not work. But I tell everybody, I think it is equally as important to figure out where this doesn't work, where this method just completely falls apart. Because by understanding where it doesn't work, we can figure out what characteristics make it work really well. So for example, with the counting algorithm I was explaining to you, there's some cave environments that are called pit caves, where it's like a big opening in the ground and the bats just fly out at your face, where because of all the dense foliage, we can't actually get accurate counts from thermal videos. So we're working on having best practices for how to place the thermal cameras. So by understanding, in the same vein with the acoustic method, by understanding where, when, what species this just absolutely isn't working, it can either inform better ways of acquiring the data, or we can just say, okay, this is the limit. Like this in theory works great, but it's only useful in a very limited range of application. We know this works great for bats, right? And if I only find out that this works great for bats, I think that's a huge success. If we find out that it's like, not so great for frogs and not so great for penguins, well, at least we tried and we had fun. And that's kind of why we do this, right? We like to have fun with this. Ben says some penguin species should be a great candidate for this approach. Oh, awesome. Great. Cool. Love it. Love it. Love it. Love it. Monitor bird migration. Yeah, absolutely. Okay, someone asked for bats. Are your recording times bounded by the start and end of the emergence periods? Because we are confident that there's little to no re-entry at that site. But for other species recorded in place, what is the time bounding approach? So if I understand the question correctly, it's how do we filter out when the bats are coming back into the cave? Because bats leave and then they come back in, and there are some situations in which they are swarming. If I'm understanding Gio's question right, so the idea of time bounding. So for most of the bats, there's a very definitive start and ending phase of emergence. Some situations, especially for the hibernating bats and what's called the spring emergence, there is no set period. So just like how we're using our thermal counting algorithm to figure out which direction the bats are going, down the line with this, I want to explore using directional microphones pointed in opposite directions, one pointing towards where the bats would be flying if they were leaving the cave, and one pointing to where the bats would be coming if they were re-entering the cave. And then just by doing energy subtraction between the two, you could account and control for any bats that are potentially doing this vortexing or coming back in. So that's kind of the idea that I have for that, Gio. Great question. Destiny has a question about the BAT software. Send me an email, Destiny. I'll send you a link. We're going to be having a publication on that real soon, but it is currently freely available in the beta testing stage, and I'm happy to explain how the software works. Someone asked, has anyone tried ASCR for penguins? I don't know what ASCR is. I have no idea. I have no clue. Does anybody know what that is?
So acoustic spatial capture recapture. So basically they place multiple microphone, and by detecting the location of the animal, they like it can be used for calculating the distance. Based on that, you can estimate the population. So it can be very much very useful for frogs. Actually, this AACR package is designed for frogs only. So
awesome. Thank you. See, I knew I would get some awesome ideas to look into. I will go look that up. Fantastic. Thank you so much. And Laurel wants to find out, get outlier estimates on days when weather prompts them to pop in and out.
That sort of goes to whether you
bats, penguins, or frogs. Which taxa?
Mostly bats.
Yeah.
So if you're getting re-entry where they start to come out. It's like, ooh, actually, this is enough. Rainy days might just be low days, but you might actually. Yeah.
Well, I mean, I think that's another interesting thing too, just from the data itself, just understanding population trends and how the emerging population, I mean, when there's a thunderstorm, I've seen bats, they all leave the cave, but then 20 minutes later, they turn around and nope, right back into the cave. So getting information on just how they're responding in general, I think would be great. Rain problem for the energy-based detection. Well, the short answer for that is we haven't yet recorded in the rain, but I'm sure it will be. Thank you, Marcelo, for reminding me of a lot of the challenges with field work. Yes, I'm sure rain detection. In Antarctica, wind is going to be a huge issue. However, the penguin vocalization should be spectrally isolated from the wind noise. So by just doing some bandpass filtering, we should be able to isolate that out.
Thank you so much for your time, Laura. I'm cognizant that we're coming up on 11:30. I actually have another meeting that I have to hop into, unfortunately. But this is really interesting to see, especially the taxonomic diversity of it. It's fun to, as you say, imagine the cases where it will work well and the cases where it won't work well. I can see fish or things like that being potential candidates.
Yeah, absolutely. Well, thank you so much, everyone. And I mean, if anybody's has any other, based on your experience, if you say, oh, have you considered this? Have you considered this? Have you considered this? Please, please, please email me, because, or find me, you can find my contact information on my website, because if there's things I'm not thinking about that I should be thinking about, this is why I reach out to the community. So thank you, everyone.
Hope to see you on 2 weeks from now. Take care and have a great week.